This video is to show you one way that you can dissect a crayfish and to point out some of the features that you should look out for along the way. Some of these features are generic to all crustaceans, some of them are more specific to crayfish. Now this here is a signal crayfish. That's Pacifastacus, leniusculus. They look very similar to our native crayfish, which is the white-clawed crayfish. Now white-clawed crayfish are a protected species, so please do be aware that in order to protect our native crayfish, that crayfish can only be collected from the wild by people who are licensed to do so. This individual here is a male, and I'll go into how I know this in just a bit. But first I want to have a little look at what we can see just from the outside. So the crayfish body is made up of many individual segments. We can't see all of the individual segments because some of them are fused together. The crayfish body is considered as having two distinct regions, or tegmata, which are the cephalothorax, which is this part here, and the abdomen, which is this part here. So firstly, here in the head area of the cephalothorax, there are 6 fused head segments, which we can't actually make out individually because they're hidden by this head shield. The head shield extends right the way forwards, ending at the tip here, which is called the rostrum. And this whole head shield is formed by an infolding of the integument from just behind the head. So it extends forwards. Now behind the head we have the thoracic region, which is made up of 8 thoracic segments, which again are fused together, and these are also fused with the head segments, so they actually form one part or one tegma. Now, like for the head, we can't make out the individual thorax segments because they're behind this, which is the carapace. And like the head shield, the carapace is formed from an infolding of the integument from just behind the head, but this time it extends backwards and also down the sides of the crayfish. The second posterior tegma of the crayfish is this abdomen, which is made up of 6 segments, and each segment is enclosed between 2 plates. So you have the dorsal plates on the back here called abdominal turgor, and you have ventral plates underneath, which are called abdominal sterna. Now the whole crayfish is covered by this hard exoskeleton, which is made up of these hard protective plates. I've already mentioned the head shield, the carapace, and the turgor and the sterna. Now these are all secreted by the underlying epidermis. This exoskeleton is made up of chitin, which is a protein, just like the cuticle that you get on the outside of insects, but in crustaceans it's also impregnated with calcium carbonate, so it's much harder. And also, unlike insect cuticle, it's not waterproof. The exoskeleton protects against predators, but one drawback of having this exoskeleton is that it can't expand as the individual gets bigger. So periodically, the growing crayfish will have to molt and grow a new exoskeleton. There's also inside of the crayfish a calcified endoskeleton, which just like our skeletons provides internal support and sites for muscle attachment. So looking more closely at this head end, you can see that the crayfish has 2 compound eyes that are out from the body on stalks. If you look closely at these eyes with a hand lens, you might be able to make out that these eyes are made up of many individual light-sensitive structures, which are called ommatidia. Okay, now let's flip this crayfish over and look at the ventral surface. Now virtually all of the segments in a crayfish give rise to paired appendages, which we can distinguish. Now these are really interesting because they're all adapted for slightly different purposes. So the first 2 appendages here, which are on the first head segment, are called the antennules, which are these smaller antennae just here. And these longer ones here are called the antennae, and these come out from the second segment of the head. Both of these antennae are involved in sensing the environment. Now if you look closely, you'll notice that they're covered in hairs. These hairs contain chemoreceptors which send information to the brain, which is located inside of the head, and they help to detect food, detect sexual partners, and detect predators. The next run of appendages down to and including this first limb, which has these huge pincers here, are all involved in food processing, such as collecting, manipulating, or breaking up the food to be ingested. So let's run through these in turn. So the mouth is here, and you can see that it's surrounded by these 2 mandibles. These mandibles arise from segment 3 in the head, and they function in the breaking up of food. Then next to the mandibles you have the maxilles, which you can just see there. Then you have a pair of maxillae, and then you have 3 maxillipeds, 1 ending that sort of long structure there, 2, and 3. The third maxilliped is quite large. And these all manipulate food in some way. Then you have this first limb, which you can see is highly modified with these huge pincers, which are known as chela, and these are used to tear off food and bring it up to the mouth, and they help steady the food whilst the mandibles work away at it. They're also very handy in self-defense. Now, moving backwards along the crayfish, we then have these 4 jointed walking legs. These 4 are all involved in locomotion. If you look very closely, you can see there are numerous hairs on the tips of the walking legs. These contain chemoreceptors, which are used to identify food in the substratum and on the mouth parts. And the fact these walking legs are all jointed means that these crayfish can move quite fast. Then on the abdomen, you'll see these rather feathery type appendages, which are known as pliopods, or you may find some people call these swimmerets, which refers partly to their function because they are used in swimming, but in the female they're also important in brooding the eggs. In just a bit we'll just see how they differ from the males. So this is a male. Finally, at the end here, we have the uropods, 1 on each side, 1, 2, and the telson in the middle, which you can see together with the uropods forms this tail fan, which is important in swimming, and you can see in the centre of the telson you have the anus. So looking back at these pliopods, how can we tell the difference between a male and a female crayfish? Well, we can see on the male they actually have 5 pliopods. 1, 2, 3, 4, 5. That's 5 pairs of pliopods. And the first 2 are highly modified. They're larger than the posterior 3. These are copulatory pliopods, and they help to deliver the spermatophore to the female. Another feature of the male is that we can actually see the sperm ducts at the base of the 4th walking limbs. And it's from these sperm ducts that the spermatophore leaves the male and it's deposited on the female. So here I have a female crayfish. Now this specimen is smaller than the male. That is just because this specimen is smaller. The females can be as large as the males. They can be larger. So just because it's smaller is not an identifying feature of the female. Actually, this female, you can identify it because you can see that it has some oviducts here at the base of the 2nd walking limb, and it's from here that eggs will be shed. So the spermatophore from the male will be deposited around this region here, eggs will be shed from the oviducts, fertilisation is external, and then the female will stick the eggs on her pliopods, which you can see here. This female has a whole mass of eggs all on her pliopods. Here they will be brooded, the crayfish develops inside of the egg and actually hatch as miniature adults. Now I'm going to remove those eggs so that we can actually see the difference between the female and the male pliopods. So I'm just gently prising them off the pliopods. They're quite well stuck on actually. And then when the crayfish is alive, she would be wafting these pliopods to ensure that the eggs were well oxygenated. So we can now see the female's pliopods, and you can see that there are 4, whereas the male had 5. 1, 2, 3, 4. And these are all more feathery than the males. So we're now ready to start dissecting. So I've got my male crayfish again. So to begin with, I'm actually going to concentrate on just removing the outside of the crayfish, just 1/2 of it, because this makes it a lot easier to handle the crayfish. The first bit I'm going to do is just to remove this top section here. So I'm going to use my scissors, I'm going to cut around here, then I'm going to come back and cut around here, and I'm going to remove that top section. Don't go in too deep with your scissors, otherwise you will damage the underneath of the crayfish. Now, I'm not going to just whip that off. I'm going to be careful. I want to peel it away and make sure any structures that are stuck to the top of this are peeled away gradually, rather than whipping them all off, and I could risk tearing the underlying structures. I'm just going to scrape it like this as I peel away, and I'm looking inside the crayfish as I do so. It makes a much cleaner dissection. So I've just removed the top section of the carapace and the head shield, and we can see a number of structures. we can see at the top here we have something called the gastric mill, which is part of the foregut. And this is made up of two regions. We have the cardiac stomach, which is nearer the head end, and the pyloric stomach, which is towards the posterior. And this actually contains sclerotinized teeth, which help to mechanically break down the food. Now, surrounding the gastric mill, we have a number of muscles which help to operate this grinding mechanism. We have the cardiac muscle up here at the front, and we have the pyloric muscle here at the back. At the sides, we could just about make out some mandibular muscles, which obviously operate the mandibles. Here, this structure which is red in my specimen is the heart, and usually you can make out there are two holes in that heart called ostia. The crayfish has an open circulatory system. There are some vessels leading to and from the heart, which we can make out. They're red, just little red streaks. The heart is contractile and it helps to circulate the blood around the body. Now, immediately underneath the heart, we can make out the gonads. In this case, it's the testes. We can just see it poking out here. This more orangey bit here is digestive gland, so we can see testes, we can see a bit of digestive gland. I'm now going to remove the whole side of the carapace. To do this, I'm going to hold the crayfish on its side, and I need to cut around here. And then I'm going to peel back this segment to reveal the structures underneath. So I'm not going to rip off this whole side, I'm actually going to scrape it away. And to do this, I find it easier to use the scalpel. So just get the scalpel underneath where you've cut and just cut away at those tissues that might be tethering that carapace to the rest of the body. Pull that away. So I've removed the whole side of the carapace, and what we can see here are the gills, these feathery structures here. This is the site of gas exchange, and the feathery structure increases the surface area available for the gas exchange. The gills arise from the bases of the 8 thoracic appendages, so that's the 3 maxillipeds, and then you have the 5 limbs. The part that actually originates from the base of the appendage is called the epipodite, and you should be able to find all 8 of those. So to find those, if you you can look at the gills in more detail just by pulling them away from the side of the body and carefully working out where they all originate. I'm not going to identify all 8 of them because my purpose is just to show you the dissection. So I just want to show that there's the 8th one on the 8th segment. You can see it just rises further up than the rest. So I'm going to lay the gills aside like so, because I'm actually going to remove now the wall of the thorax, and I'm going to use my bowstring scissors for this. I'm also going to remove a bit of the head shield. So take care, don't cut too deeply into the crayfish. Just use your bowstring scissors just to take off that the thoracic wall, side of the thoracic wall. And you should be cutting quite close to the bases of the thoracic limbs, the appendages. It's really like with anything, when you're pulling something away from the main wall, try and just gently prise it away rather than just ripping it away. Move the underlying structures underneath so that they don't come away with the wall. That's the side of the thoracic wall removed. I also now need to remove this head shield. Okay, so what we can see here, filling up most of the body cavity, is this bright orangey coloured digestive gland, which some people also refer to it as a hepatopancreas. It is in fact one of 2 blind-ended sacs, or the proper term is diverticular. So there's one this side, there's one on the other side. And they're part of the midgut, and they're made up of lots and lots of fine tubules. This is where digestion and absorption takes place. It also functions as a storage organ, storing some fat and some glycogen. Now there's a snotty green coloured gland just inside the crayfish here. On my specimen here, it's not as obvious as it may be on yours. I can pull it out from just behind the head. It's just here. This is called the antennal gland, or some people call it the green gland. And this basically does the function of a kidney. So it's the organ of excretion. And you can see that it actually lies in front of the mouth in the segment that bears the antennae. It's where excess salts and water are got rid of, and also nitrogenous waste in the form of ammonia. Now, I want to see the digestive system. In order to do this, I need to remove the turgor on the abdomen. I'm going to use my scissors. I'm going to make a cut down one side like so, and the other side like so. Then I'm going to peel this back, but taking care so I don't bring all the extensor muscles. That's those that will actually extend the tail up. Make sure they don't come away as well. You can see the extensor muscles just coming away. So you can see if I pulled these, the extensor muscles would come away. We can just see the gut poking through there, which is why I just want to take care really of taking this off rather than just ripping it away. Okay, so we've removed that in order to see some of the digestive system. We can see the hindgut here. If I now peel away carefully some of the extensor muscle, it will reveal the hindgut in the abdomen. So we have food taken in through the mouth. It will come through the foregut, which is this gastric mill. We're going to remove that in just a bit. The food that is to be digested will be diverted towards the digestive gland when it enters the midgut. And the rest of it will be shunted along the alimentary canal. We'll be able to see that. Then it will enter the hindgut, which of course terminates in the anus. Move aside the mandibular muscle. Put that to one side in order to be able to see the gut a bit more clearly. Moving aside some of the testes and the digestive gland as well. If I lift it all up, we can actually make out the digestive system underneath. Running along the top of the gut, we have a blood vessel. I'm now going to remove the gastric mill. I'm really intrigued by the gastric mill because of these so-called teeth. I really want to see these teeth. So make sure those mandibular muscles are well out of the way. Then I'm going to cut close to the midgut. I'm going to cut through the cardiac muscle at the top here. And I'm going to cut through the pyloric muscle, which is just here. Now be careful to keep an eye on the orientation of your gastric mill as you remove it. I'm going to use my blunt forceps. I don't really want to damage the structure. And just cut through the esophagus at the bottom. That will be keeping it in place, so we can fully remove it. And we're going to place that onto a petri dish. Okay, so just to orientate us, this is the cardiac muscle I've cut through. This is the pyloric muscle I've cut through. And if I turn it over onto its dorsal surface, we'll have the esophagus around here. So the first cut we want to make, we want to open this up and make it flat. So basically we want to cut it open from the esophagus down to the midgut. So the whole thing will open out. So just to remind ourselves, this is the cardiac muscle side, this is the pyloric muscle side. We want to pin that. And what we can see now inside of the cardiac chamber, we have these teeth, which are sclerotinized. So food would pass through the gastric mill in this direction. It would go into the cardiac stomach first, which is lined with these numerous ossicles, which are teeth, which will help grind up the food. And the ossicles have evolved from the cuticle, so there's cuticle lining the foregut. There's also cuticle like that lining the hindgut as well. Then it will enter the pyloric stomach, which is this part here, which serves a more assortative function, directing the digestible matter to the digestive glands, whereas the larger indigestible matter will be shunted along the midgut to the hindgut. So there's some filters in the pyloric stomach just for this purpose. So now comes the messy part of the dissection. I want to see the nervous system, which actually lies ventrally in the crayfish. So I need to remove the overlying structures. So I need to remove the orange digestive gland, which you'll see is very, very extensive. I need to remove the gonads, so in other words, the testes, the heart, and I need to remove the antennal glands. And what I couldn't show you before was the midgut that runs all the way through. So the digestible portion of food will be diverted to the digestive gland. The indigestible matter will be shunted through this very straight midgut region, which will lead straight into the hindgut. Another feature that I can see in my dissected specimen is the esophagus, which led to the gastric mill. Now, to reveal the nervous system, we actually also need to remove all of this side as well. So just you remove that in exactly the same way as you did for this side. So I've removed all the corresponding tissues on the other side. I've also removed the gut. Now, have a look at these mandibular muscles. Look how big they are. It just goes to show how strong the muscles of crayfish must be. So crayfish are omnivorous and they really will eat anything. They eat shellfish, worms, plants, insects. And they'll also feed on decaying and dead matter, so dead plants, dead fish. So they really will eat a wide range of food. Those are the mandibular muscles. So we want to have a look at the nervous system. So they have a ganglionated nerve cord which runs ventrally. We can't see this yet because it's underneath the overlying tissue. So it's underneath all these tissues here. It's underneath these flexor muscles here and here. So we need to use a scalpel to cut away these flexor muscles. It is quite tough and they are attached quite deep. So they're attached to quite deep on the outside of the abdominal sterna. So I need to cut away there. And then I'm going to cut away this part here as well. But just bear in mind that the nerve cord runs centrally to the crayfish, so just make an effort not to damage the structures that run centrally. So as I'm peeling it away, you can see it's quite tough. The nerve cord, I can just see the nerve cord in there, which is reassuring to me because now I know how deep it is. It gives me an idea of how deep I can make my cuts without damaging the nerve cord. So what we can see here, we have right at the top here, we have the cerebral ganglia. The cerebral ganglia consists of 3 ganglia which are all fused together, and this is effectively the crayfish's brain. It receives all the information from the eyes and the antennae, and it controls the movements of the anterior head appendages. Here is another important group of fused ganglia, which is the subesophageal ganglion. This is a fusion of ganglia from the mouth parts Perhaps what we can't make out so clearly here is that there is actually another important ganglia behind this, which is the fusion of the ganglia from the first 3 thoracic segments, which if you remember are the maxillipeds. So this here is sort of the centre of food processing. After this you have 1 ganglion per segment. So if you have a look closely, you can see more obvious places on the ventral nerve cord. That's the ganglia, ending in the 6th abdominal ganglia right at the end. Each of these segmental ganglia receive sensory input from the segments that they serve, and they provide motor nerves to the appendages to those segments. So you can see the nervous system of the crayfish isn't completely centralised, which is why parts of the crayfish would still move in a purposeful way if you only disable the cerebral ganglia. And this completes the crayfish dissection.